diseases. These are Down syndrome, Klinefelter syndrome, and then the Turner syndrome. So, what happens in Down syndrome? So, the Down syndrome is a genetic disorder, it's a chromosomal disorder where it's a trisomy of 21 chromosomes. Now, what is that? It's the presence of an additional copy. It's the presence of an additional copy of the chromosome number 21. Of the chromosomal number 21. So this is the trisomy of chromosomal 21 because it's the 21 chromosome. There is an additional copy. So it becomes triplet. Initially it was doublet. Initially it was doublet. As you can see this in this diagram, you see a representative figure showing an individual infected with a Down syndrome and the corresponding chromosomes of the individual. You can see in the 21st in the 21st position, you see it should be doublet, but it becomes triplet. That is an additional copy of the chromosome number 21. So this was the Down syndrome. Now the Down syndrome, okay, and was first described by Langdon Down. The name itself, Down, okay, Langdon Down stated the Down syndrome in 1866. In 1866. Now let's talk about some features. Now the affected individual is short. It's short statured with a small round head. With a small round head, furrowed tongue, then partially open mouth. Partially open mouth is the symptom for Down syndrome. Now the palm, that is the hand. Palm is broad. It's broad. Usually it's not broad. The disease palm is broad with characteristic palm crease. It's kind of crease. It's kind of bold line on his hand. Okay, so that's what the palm crease, and palm is broad. Now physical, psychomotor and mental development is also retarded. So this was the symptoms of Down syndrome. Now let's see the actual boy with this syndrome. Now you see in the diagram, flat back of head. Usually the head is round, small round head. It's given in theory. So flat back of head. So head is also flat at the back side. Now broad flat face on the upper side. It's broad flat face. Now palm crease, you can see the palm crease on his hand. So it's a bold line on his that palm region. Now another additional thing is many loops on fingertips. Okay, many loops on fingertips. Usually the tips have the loops, but it has many loops on the fingertips. Now talking about the tongue, it's furrowed tongue, additional big and wrinkled tongue. The tongue is big, wrinkled, furrowed tongue. And the Down syndrome individual has congenital heart diseases. So it can also lead to the congenital heart diseases. So heart diseases is also involved in the Down syndrome. Now the next genetic disorder, that is a chromosomal disorder, is Klinefelter syndrome. Now what happens in this syndrome? Let's talk. So in Down syndrome, it was the trisomy of 21st chromosome, but now these two diseases, that is Klinefelter syndrome and Turner syndrome, so these syndromes are related to the sex chromosomes. Now in the Klinefelter syndrome is the presence of an additional copy of X chromosome resulting into karyotype of 47, that is XXY. So if it was male, it should be XY, but additional copy of X chromosome. In Down syndrome, it was not X or Y, it's just the additional copy of the chromosome number 21. But in the Klinefelter syndrome, it's the X chromosome that is the sex chromosome. Okay, so this was it. And in Turner syndrome, you see it's opposite to the Klinefelter syndrome. There is an absence of one of the X chromosomes. It should be XX, but it but it's the absence of the X chromosome. It becomes XO X0. So 25 with X0. A human has 26 chromosomes, right? Now, one chromosome is absent, and that was the X chromosome in the females. A female should be XX and male should be XY. Okay, so you can remember as Klinefelter syndrome is related to the males and Turner syndrome is related to the female. Okay, now see the additional symptoms in the Klinefelter syndrome. Now, such individual has an overall masculine development. However, the feminine development, that is development of breast, that is gynecomastia, is also expressed, and such individuals are sterile, that is they cannot reproduce. Now see in the diagram. This tall, statured with feminized character. You see gynecomastia, that is the breast development, and has an overall masculine development. This male has general masculine development, right? It's not that weak. Has a general overall masculine development, but along with the feminine character that shows the gynecomastia, and such individuals are sterile, that is they cannot reproduce. Now the Turner syndrome, see in the B diagram. It's a now see in the theory first. So such females are sterile. So in Klinefelter syndrome, these males are sterile, and in Turner syndrome, these females are also sterile. So such females are sterile, as ovaries are rudimentary, because ovaries produces ovum for reproduction, right? If their ovaries are not that good, it's just rudimentary. Besides other features including lack of other secondary sexual characters, example like breast and like things like that. Okay, so they also lack the secondary sexual characters, so we can say that they are the sterile, that is they cannot reproduce. Now see in the diagram, so it's a short stature. Now in the Down syndrome, it also the short stature. You see in the Down syndrome, it's also short statured with small round head, but in Turner syndrome it's also short statured, but in the Klinefelter syndrome it's a tall stature, right? Okay, so if question is asked like short stature syndromes tell, so it will be Turner syndrome and Down syndrome, and tall stature is the Klinefelter syndrome. Okay, so the short stature and underdeveloped feminine character. Okay, now figure 4.17, diagrammatic representation of genetic disorders, that is chromosomal disorders due to sex chromosome composition in humans. So due to sex chromosome composition, so these disorders are Klinefelter syndrome, Turner syndrome. While if they ask the autosomal chromosomal disorder, that goes to the Down syndrome, that is the additional copy of the chromosome number 21, that is the trisomy of 21. So this was it, and this figure shows the Down syndrome. These two figures shows the first one shows the Klinefelter syndrome, second one shows the Turner syndrome. Now Klinefelter syndrome, it's the is the karyotype of 47 with XXY condition, and in Turner syndrome you see it's the 45 with XO type of condition. While the Down syndrome, it shows the trisomy of 21st chromosomes. It's a triplet of 21st chromosome. You see in the diagram, there at the 21st position, it's the trisomy of the 21st chromosome. That is additional copy of the chromosome number 21. So this was all about these chromosomal disorders. Now, along with that, You have seen the chromosomal disorders. There is additional copy of chromosome number, additional presence of X chromosome, and in the Turner syndrome there is a absence of X chromosome. So these are like one is additional and one is less, so type of thing. So what is this? So this is the aneuploidy. What is that? What is aneuploidy? So it's a failure of segregation of chromatids. So it's a failure of segregation of chromatids during cell division cycle, results in the gain or loss of a chromosomes. So a single chromosome, if it's lost, then it's a Turner syndrome disorder. If it's gain a single chromosome, then it's a Klinefelter's syndrome. Okay, and if additional copy of one chromosome, it's a Down syndrome. So generally it's a aneuploidy is a process, just that. And chromosomal disorder is due to the absence or excess or abnormal arrangement of one or more chromosomes. Abnormal arrangement take, okay, due to absence is the Turner syndrome again, is the excess that is additional. You can see the Klinefelter syndrome or Down syndrome or abnormal arrangement. Okay, so these causes the chromosomal disorders, these three things. Now, what is polyploidy? Let's also this. You see, it's given written in here that Down syndrome results in the gain of extra copy of chromosome 21. Similarly, Turner syndrome results due to the loss of an X chromosome in human females. So its disease is related generally to females, Turner syndrome females. Now let's also see the polyploidy. So failure of cytokinesis after telophase stage of cell division results in an increase in a whole set of chromosomes in an organism, and this phenomena is known as polyploidy. This condition often seen in plants. There is an increment in the whole set of chromosomes. It means if 2n becomes the 4n, 4n becomes the 8n, there is a whole increment in the set of chromosomes, while in the aneuploidy is just a gain or loss of a chromosome, a single chromosome, right? So that's the difference between aneuploidy and polyploidy. And you can remember as aneuploidy seen in humans, okay, because these are the disorders, and polyploidy often seen in plants. You can remember like that, right? And generally humans have 23 pairs of chromosomes, while 22 pairs are autosomes, and in Down syndrome is autosome chromosome that gets additional. So that was the autosome, not the sex chromosome. And in diseases like Klinefelter syndrome and Turner syndrome, there is an additional or loss of an X chromosome, that is sex chromosome. So this was it. Okay, along with that chromosomal disorders, let's also see the sex determination in honeybee, which is a grand topic nowadays. So you just see this diagram first. Sex determination in honeybee, honey, which is very useful product. It's very sweet. Now, what's the sex depression? What's the sex determination in honeybee? Now you see the parents. First is the female. Now female is diploid, that is 32, which is 2n. Now n is equals to 16. So 32 and 16. Now male is 16. So after the meiosis in the female, that is 32 chromosomes. Meiosis, it is a reduction of the chromosome number, that is 32 gets 16. So it becomes 16. So these are the gametes. Now one of the 16th chromosomes after reduction division in female becomes the male, and the process is known as the parthenogenesis, right? Parthenogenesis. You see in the male, after the mitosis it becomes 16. So so it becomes 16. Now this male and this haploid female, that is 16 on this side, it gets fertilized and it becomes the female with the 32 chromosomes. So ultimately you have to remember that females are diploid, that is 32 chromosomes, and males are haploid, which are 16 chromosomes. Now see this naming. Who is the queen and worker? So queen is a queen, or worker are gulams. So you see the female can be queen and worker. An unfertilized egg develops as a male. They are drawn by means of parthenogenesis. As you see, this 16, one of the 16s from female goes after mitosis, that is the parthenogenesis, it becomes the male, and these males are known as drone. So these are the drone by the process of parthenogenesis. So this is the sex determination in honeybee. Now you see, the sex determination in honeybee is based on the number of sets of chromosomes an individual receives. The number of sets of chromosomes an individual receives. If it receives double N, so it becomes the female. If it receives single N, it becomes the male. Okay. This means that the males have half the number of chromosomes than that of a female. That means it has 16th chromosome, that is N. You see in the diagram 16, and females have double the chromosomes, that is 2N. Now the females are diploid having 32 chromosomes, and males are haploid, that is having 16 chromosomes. So this is called as haplodiploid sex determination system and has special characteristic features such as the males produce sperms by mitosis, which is a parthenogenesis, and this is why they do not have father. They do not have father. Remember this line, they do not have father and thus cannot have sons, but have a grandfather and can have grandsons. I repeat it again. The males produce sperms by mitosis, which is a parthenogenesis, and in here they do not have father and thus cannot have sons, but can have grandfather and can have grandsons. So this was the haplodiploid sex determination system done in honeybee. Queen, worker, these are the female which are diploid, and haploids are through mitosis, these are males, that is 16 in number, through the parthenogenesis. And this is haplodiploid sex determination systems. Okay. How is the sex determination mechanism different in the birds? Difference is ZW, right? The females are heterogametic, they are ZW. Usually they should be homogametic, that is ZZ. But in birds, it's a ZW, that is ZW, and males are ZZ. Is the sperm or the egg responsible for the sex of the chicks? Of course, egg having the ZW is responsible for the sex of the chicks, while not the sperms, because they are ZZ. So important thing is that the sex determination in honeybee, you have to remember that the females are diploid, the males are haploid. The females are 32, the males are 16. That's it. So this is the haplodiploid sex determination. So queen, worker, these are the female, and after parthenogenesis, the male, the haploid one, that is 16, becomes the male. These are the drone. And in honey, sex determined by the number of sets of chromosomes an individual receives. If it's 2N, it becomes the female. If it's haploid, it becomes the male. It's kind of that simple. See in here, I already told you that female heterogamety is seen in the birds, and females are ZW and males are ZZ. Again, I'll show you. The male heterogamety is seen in the XO or X type seen in the Drosophila and humans. and exo seen in the grasshopper. So these are the direct questions you see in the diagram. You see on the top of that, grasshopper is an example of exotype of sex determination in which the males have only one X chromosome besides the autosomes, whereas females have a pair of X chromosomes. See this diagram. Let's place this. Now, determination of sex by chromosomal differences. So these sex determined by the differences in the chromosomal differences. So both in males, humans and Drosophila, the female has a pair of XX chromosomes like us, that is human females. These are the homogametic, and the male have XY, that is heterogametic composition. Now, in many birds, these are female has a pair of dissimilar chromosomes, that is Z, W, and the male two similar ZZ chromosomes. ZW females have and males have two similar ZZ chromosomes. See the diagram. So in the first diagram, you see the male on this side, X straight chromosomes, Y bended chromosome, and then the XX on the female side. Arrow tops. If the arrow is at the top, then it's male. If arrow is at the bottom, it's the female. Now in the second diagram, you see the male Drosophila is short than the female. Female is kind of large because she eats a lot. You can remember like that. And in the chickens, these are the birds. On this side you see Z, it's a male. On this side it's a female because W is bended. Let's also see these symbols used in the human pedigree analysis. You see on the top place, these are the males. These shown by the rectangles because males have the corners. No, males do not have the corners, but males are complicated, so this is they having the corners. Now females, they are circular. You can remember like that because females that don't have that much complications. So you can see females are circular, males having the corners. Now sex unspecified because if you take the male and just rotate it like that, and that becomes a sex is unspecified, right? It's no more the male, right? Now in the fourth place, you see take that male and take that female and take that rotated male, that showing the sex is unspecified. Now darken that color, take the crayons and dark them, and that becomes the affected because they are now not no more pure. Okay, so they are dark, so they are affected now. So these are the affected individuals. Now on the fifth position, you see take that male on top and take female and mate them, so this is a mating. Now on the sixth position, take that fifth position and make a rod between them, and that's the mating between relatives. That is a consanguineous mating. Take an example of relatives in your family, so you can remember like that. So that's the male and that's the female between two rods. Now So the general marriage is the single rod, right? You can remember, like if it's in the relatives, it can be double rod. Now parents above and children below, you see the parents, that is a rectangle and circle at the top, because parents your everything. So that's why you are in here. So parents are always at the top, so parents above and children's below. And even even in the children, if you have the older brother, that means your brother should be first. So you see, in order of birth, left to right, you see this. In this diagram, the female is older, so that becomes a king for this male, on the male which is on the right hand side. Okay. Now parents with male child affected with disease, you see the rectangle and circle, they meet. So that's a normal mating, and there is an offspring. So parents with male child affected with disease, so parents are at the top. Now it's darkened in color, so male child is affected. Now at the third position, I already told you that if you rotate a male, which is a rectangle, if you return 5 inside that box, so it becomes, so at first it was the sex unspecified, but 5 unaffected offspring, sex unspecified, but we return the 5, so 5 unaffected offspring. Sex is still unspecified, so this is why offspring, so 5 unaffected offspring. So this was it. So these are the symbols used in the human pedigree analysis. Let's talk about the sickle cell anemia. The defect is caused, see this bottom line, the defect is caused by the substitution of glutamic acid. It's an amino acid, that is glue, by valine, that is val, at the sixth position of the beta globin chain of the hemoglobin molecule. Remember that hemoglobin molecule, it is a pigment, it is a respiratory pigment that carries oxygen and carbon dioxide. And this hemoglobin having the alpha chain and beta chain. So in this disease, the beta globin chain is affected. At the sixth position, glutamic acid is substituted by the valine, that is val. So this was the sickle cell anemia disease. Now take into this codon. What happened? The codon is made up of, you see, for example, GAG to GUG. So GAG is a codon. Now let's see the theory. Make the focus. Okay. So the substitution, it's not elimination, it's not addition, it's a substitution. It's a substitution. A new employed is replaced by an AI. So that's the substitution. The gap is filled by someone else. The substitution of amino acid, that is glutamic acid, in the globin protein. Remember that hemoglobin is also a protein, if I'm right. Results due to the single base substitution. Now codon is GAG. G is guanine, if I'm right. A is adenine, G is guanine. So G is base, A is base, G is also base. So this base, a single base substitution at the sixth codon of the beta globin gene from GAG to GUG. So A is replaced by U. So this condition is the sickle cell anemia, that is GAG to GUG. Now, but it says the mutant hemoglobin molecule under no above line. The substitution of amino acid in the globin protein results due to the single base substitution sixth codon of the beta globin gene. It says gene, so it's gene, beta globin gene from GAG to GUG. Now the what happens after this? It is disease happens. What happens? Now it becomes mutated low hemoglobin molecule undergoes polymerization and under low oxygen tension, causing the change in the shape of the RBC from the biconcave disc. RBCs, human RBCs are biconcave in shape, so biconcave discs to elongated sickle cell structures. So if it says tell the disease that undergoes the RBCs that change shape from biconcave discs to elongated sickle cell like structures, and they show the polymerization under low oxygen tension and having the single base substitution at the sixth codon of the beta globin gene or chain from GAG to GUG, that is glutamic acid to valine, and also so the disease would be the right answer would be the sickle cell anemia. Now again in here, so first thing is that the sickle cell anemia was an autosomal link recessive trait. It means it's not sex link recessive disease like hemophilia, color blindness. So these are the sex link recessive disease, but they are sex link, right? Sickle cell anemia is autosome, which are 22 pairs. Now, three type of combinations are present. You have to remember that. So first is HbA. So it's a normal condition. Now HbS, it's an carrier, that is unaffected condition. HbS is the affected conditions. Okay. Now the important line is, you see, the heterozygous which are carrier. Now they carry, they are not affected completely, but they just carry, they have the genes. Now these heterozygous, that is HbA HbS, A is normal, S is affected. Now individuals appear apparently unaffected. They look unaffected, but they are carrier of the disease, as there is 50% probability of transmission of the mutant gene to the progeny. That means HS gene is affected, right? So it can transmit, that gene can be transmitted to the progeny and thus exhibiting the sickle cell trait. Now look at the top line. So sickle cell anemia. So this is an autosomal link recessive trait. Why it's recessive? In simple words, HbS is affected for sure. Now this is the condition is a homozygous, right? So you can see that. So you can say that it's a recessive, right? It's shown, this affected is shown in the homozygous recessive condition. Just remember that like that. So this is an autosomal link recessive trait that can be transmitted from parents to the progeny when both the partners are carrier for the gene or heterozygous. It means to occur this condition, to occur the sickle cell anemia, both the parents, male and female, must be carrier. And carrier condition is HbA HbS. Normal condition is HbA HbA, and affected condition is HbS HbS. Now HbA HbS is not affected. HBS is not affected. HBS is affected. This is kind of recessive, right? Now, the disease is controlled by a single pair of allele. A gene has two alleles, two alternative molecular forms, that is HBA, HBS. Now, take just think like that: tall and dwarf. Capital T is tall, small t is dwarf. Just like that. HBA is capital T, HBS is small t. One is affected now, one is recessive, one is dominant. So out of three possible genotypes, only homozygous individuals for HBS show the disease phenotype. This is a physical appearance, that is the external appearance. In a heterozygous, that is HBA, HBS, individuals appear apparently unaffected, but they are carrier of the disease, as there I already read that line. Now the remaining portion left is this diagram. So figure 4.15. RBCs are biconcave in shape from the starting, but it becomes the elongated sickle-like structures. You see, it's kind of elongated. Now the normal HBA gene, gene, it's kind of DNA that has this double standard. So first strand you say GAG, a second strand CTC. See, now this DNA ultimately will form so we have to make the protein, and protein is a hemoglobin molecule, you see. So this gene forms the mRNA through all this like transcription. Okay, so CTC forms the GAG. You know the cytosine and guanine. So these are the nitrogenous bases. Thymine, and remember that in DNA is the thymine is present, in RNA is the uracil is present. Now we want the mRNA. Okay, so C pairs with G with how many bonds? I think it's 3 bonds, right? So 3 bonds, hydrogen bonds. So C pairs with G, then T pairs with thymine, pairs with adenine, C pairs with G. So this becomes the GAG, and which represents the glutamic acid. That is the HBA peptide, which is a normal peptide. Now, so in the second diagram, you see it becomes a sickle cell HBS gene. Now, gene, which is part of DNA, it has the 2 strands. Okay, so first strand is the GTG. In the second strand is the CAC. So take CAC. So this strand will give the mRNA. So C pairs with G, A pairs with U, C pairs with G. Why it's U? Because it's an mRNA. mRNA have no thymine, so it's replaced by U. So GUG. Now this mRNA will form the protein. However, this is not the correct globin molecule, but mRNA is doing his work, so it forms the valine by mistake. Now it becomes HBS peptide. Initially it was HBA peptide, which is the normal, but it becomes the HBS peptide. So this is how, and the question can be asked from this. CTC, it will ask, the normal HBA gene has these 2 strands, GAG and CTC. You tell the mRNA, so it will be GAG. On this side, sickle cell HBS gene can have GTC, CAC. You tell the GUG. Kind of confusing, right? coding thing, base pair coding complementary. This can be asked. Now figure 4.15. So micrograph of the red blood cells and the amino acid composition of the relevant portion of the B chain of hemoglobin. Of relevant portion, that is wall, his, live, through, pro, glue. This never asked but can be asked. Wall, his, lu, throw, pro, wall, glue. Remember this HbA peptide. Okay. So portion of B chain of hemoglobin now. From a normal individual, you see the normal individual. It has a wall, his, lu, throw, pro, glue. And from an individual with sickle cell anemia, you see the sickle cell anemia, that is HbS peptide, which has the sixth position. Valine is substituted. Valine is there. Glutamic acid is substituted by the valine at the sixth position of the beta globin chain of hemoglobin molecule. At hemoglobin, it is a respiratory pigment, and pigments are mostly the proteins. So this was all about the sickle cell anemia. Let's see the polygenic inheritance. So the started from the Mendel, George Mendel, but sadly he never knew that he was a great scientist until he died.